In order to make a vaccine, so you first have to extract disease from an animal or a human. So for instance, with measles, they got blood of somebody who had measles, and they incubated that through a series of cell lines, because you can't just inject live measles into a person. You first have to do what they call attenuating it, which is to make it less virulent. So in order to do that, you pass it through different cell lines. So some of the cell lines that they pass this measles through are human cells, amniotic cells of human beings, chicken cells, monkey kidney cells. Then after they do that, they have to multiply it so that they can make massive amounts of vaccine. In order to multiply it, they'll multiply it on all different kinds of cells, like something called a maiden-Darby kidney. This isn't for measles, but for flu shots, for instance, maiden-Darby kidney cells, which are Cocker Spaniel kidney cells that have been made tumorigenic so that they multiply rapidly. Now, part of those cells go into the vaccinee. Fibroblasts from fetal lung cells, which came from an abortion in 1966, are some of the other cells that these vaccine cultures are grown on. There's a myriad of monkey kidney cells are still used today, and they've been used for a very long time. So all of these things, all of these animal cells, animal DNA, are part and parcel with a vaccine, and it's even listed on some of the package inserts. So you can imagine that different diseases can be picked up along the way, and that has happened in the past. There have been monkey viruses that were finally, after 30 years, acknowledged to have been causing tumors in human beings, been heavily associated with tumors in human beings. In addition to that, there are various unknowns that can't be picked up during testing, because if you don't know something is in a vaccine, you can't test for it. You have to have a specific test. Things have been found by third parties. Viruses, stray viruses, have been found by third parties in vaccines.